We have 32,000. I checked with Martin, 0.41 is the expected yield. That doesn't mean he didn't do the reaction. Okay,
so the answer is no.
So the answer is an incredibly super large. Okay, so anyway, it's not actually 0.049 grams.
I got
0.00049.
Like, I want to say
yeah. It's 2 zeros or 3?
3.
Yeah, I have that. I have that.
We have 32.
Rosa, did you check your yield?
So, the original that's my Because we actually get
cucumbers like water.
Because it ends up, you get 18 miles. You just multiply that by, it's a 1-to-1 ratio.
All right, good morning. How's everyone doing?
I'm just hungry.
You're hungry? I'm hungry too.
Okay, cancel class my table.
Today's a really important day. Today's the day you guys get to bring all your questions before the exam on Friday. You guys know there's an exam on Friday, right?
Yeah.
Okay, making sure. Okay. Briley? Brianna A. Drew? Rhea? Henry? Henry? Anna? Nick? Abby? Hannah?
Here.
Rosa?
Here.
Matt? Jillian? Mia? Annie? Brianna Hill? Brandon? Sadie?
Here.
Jessica?
Here.
Jesse? My God, every time I say Sadie. Jesse?
Here.
William? Marissa? Anna? Sophia? Matthias?
Here.
Colin? Olivia?
Here.
Braden?
Here.
Amber?
Here.
Katie? Katie? Kennedy? Ben? Ava? Ava? Kendall?
Here.
Emily?
Here.
Lauren?
Here.
Jen? Isabel?
Here.
And Corwin? All right. Who is going to be taking exam 2 today? Okay. Kendall, right? Kennedy, sorry. So close. Rosa. Kennedy and Rosa. Anyone else? And what time are you guys coming?
I have to take another one at 3, but I can come after that.
You can come at 2 as well. Can you do 2?
Okay. Yeah, I can do that.
Kennedy at 2, Rosa at 3. Just making sure I had it right in my notes. Okay. Okay. So for today, guys, and this is being recorded, so you guys can watch this later. But we'll kind of let you guys lead the way. Just tell me which chapter you're referring to. You guys can ask any questions and then have them up here, and then yeah, try to also give me an idea of the slide number, and we'll go from there.
Could you go over and explain the lac, Y, lac, Z, and lactose?
So the lac operon basically is, an operon is a segment of DNA that has genes on it that have a function. And so the lac ZYA operon encodes 3 genes, Z, Y, and A, that basically produce enzymes that can break down lactose. So the enzymes, the 2 that you should know, are beta-galactosidase and lactose permease, so lac-Z and lac-Y. You should know the functions of both of these. Lac-Y is the permease, the protein that allows lactose to get into the cell. Lac-Z is beta-galactosidase. It's the enzyme that cleaves lactose into galactose and glucose. Lac-I is another part of this operon that basically, comes before the promoter for the lac-ZYA, so, and it has its own promoter, and basically lac-I is where the repressor binds to repress the lactose operon, because if something is on, it also needs a mechanism to be turned off too. So that's what the lac-I region is for, and the lac-I repressor is the protein that will bind there to repress transcription of these genes that break down lactose. So in a normal paradigm, let's go up 1. This is a lac permease, lac-Y. It allows lactose to get into the cell. And then if there's high levels of beta-galactosidase, the lac-Z, then lactose can be broken down into the two components, galactose and glucose. If there are low levels of this enzyme, lac-Z, then lactose is converted to an intermediate called allolactose. And so allolactose has a role later on that we talked about, but essentially you guys should know, what the functions of lac-Z, lac-Y, with high levels versus low levels of beta-galactosidase, lac-Z, what would happen? Would lactose get converted into allolactose, or would it get converted into galactose and glucose? So the other thing here with allolactose is that when beta-galactosidase cleaves lactose into allolactose, this intermediate, because it's at low levels, then the allolactose serves as an inducer molecule that will basically bind to that lactose repressor, and then this will pop off, and then you can get transcription of more LAC-Z because the levels were low to begin with. So this is triggering the cell to then make an intermediate, which then serves as the inducer that can then pop off the repressor, and then it can make transcription of LAC-Z, LAC-Y, and LAC-A. But you guys don't have to know LAC-A because we still don't know the function of it very well. Does that make sense? Wow. Yeah, and so some other things that you guys should know. So I wrote the exam, so I'm trying to help guide you to know exactly what to study. You should understand, what happens, when there's two things present in the media versus when there isn't two things present in the media, some of these terms. You should know what dioxide growth is, and catabolite repression. And understand, how glucose will repress the LAC operon. Inducer exclusion is something you guys should know in this context. So, yeah, the LAC operon is one that you guys should know very well.
Can we go over anabolic and catabolic pathways?
Yeah, absolutely. So let me try to break this down a little bit. What is going on? One of my pet peeves is when people don't erase it completely, but it's okay. So, anabolic. Anabolic means to build things up. Basically like biosynthetics, or you're basically making a molecule. Catabolic means to break down. So in anabolic pathways, things that we want to make are things like amino acids. Things that we want to break down are sugars to make energy in the form of ATP. So if you're wondering if it's an anabolic or catabolic pathway, the first thing you should ask yourself is, what is the molecule? Is it a sugar or is it an amino acid? Is it an amino acid? It's going to be an anabolic pathway. A sugar, sugars end in O's. So lactose. So the lactose operon. Which other one did we talk about that ends in O's and should be a sugar that you can break down? Arabinose. We talked about the arabinose operon. We know glucose is a sugar, but we didn't talk about glucose operon. We talked about an anabolic pathway of an amino acid. Which amino acid was it? Tryptophan. Now, another thing that you guys should understand is, some other little intricacies within these operons. So the arabinose operon has these dual regulatory functions. So it can act as both a repressor and an activator. But the thing I wanted to say here is one of the things that is important is anabolic pathways, they're biosynthetic. You're building things up. Catabolic are degradative. You're degrading things or breaking them down. And so the repression of them is different because in an anabolic pathway, everything that you want to make, you also have to have a mechanism to repress it. And so repressing anabolic pathways is important because basically the end product, which is the thing you want to make, the amino acid, it serves as a co-repressor. So that co-repressor basically activates the inactive APO repressor and allows it to repress the transcription of meat. Because if you have a lot of that amino acid in the cell, then there's no need to make more. So that molecule, that amino acid, will serve as a co-repressor to repress the pathway while there's a lot in the cell. And when the cell runs out, then that co-repressor falls off, and then it can then make the transcription of those genes that can build up that amino acid. So you should know, how the repression of the tryptophan operon works. Does that make sense? Yeah, and then you'll go.
Can you go over the two-component signal transduction pathway?
Yeah. So this is all, so everything else that we're talking about here, when we're talking about the DNA, the mRNA, induction, repressors, activators, this is all in the cell. The two-component signal transduction system is how a cell senses the external environment. So you have two components, which are the sensor kinase and the response regulator. You should know this. The sensor kinase is in the cell membrane, and there's an environmental signal that binds to it and activates it. A lot of this happens on its own. It's auto-induced. So environmental signal binds the sensor kinase. This causes it to activate itself by ATP autophosphorylation, which just means that it happens on its own. And then once that happens, that phosphate group then can be given to the response regulator. And then once it's phosphorylated, it can bind to the DNA on the operator region, and then you can either activate or repress the genes. So this is just a very general system. I didn't give you examples of this. This is just the general, here's an environmental signal, we're not specific here. Binds the sensor kinase, triggers ATP phosphorylation, response regulator takes that phosphate group, and then will either regulate, or will either activate or repress the transcription. Anything else on this? Yeah, what's your question?
So I was just gonna ask, so in the lytic cycle, don't they kill all the bacteria? And in the lysogenic cycle, is that what it's called?
Mm-hmm.
So do they just not touch the bacteria at all because they want the bacteria to be able to replicate itself so the virus gets replicated with the bacteria?
Yep, that's exactly right. They don't not touch it at all because it's definitely integrating its genome in there, but it just allows the bacteria to replicate itself. They don't harm it. They don't lyse it. So it's just kind of it stays quiet in there, and it just uses the bacteria's replication to replicate itself.
And then in the slow release, they keep it fairly alive.
Yeah, because the virus is replicating itself inside the bacteria, and it's doing it by using the bacterial cell's machinery. So the bacteria starts to stay alive, but it's like a zombie, it grows slowly. But the difference between the lysogenic and the slow release is that in the lysogenic, the virus isn't using the cell's machinery to replicate itself. It's kind of piggybacking off of the bacteria replicating itself, so it's also being replicated automatically. Whereas in the slow release cycle, the virus is using the bacterial cell's machinery to replicate itself. And so the bacteria is also using it to replicate itself, but it's happening slower because it's being used by the virus. Make sense? Mm-hmm. Yeah.
Can you go through the arachidonics and again?
Yes. So the arabinose operon, arabinose is a sugar, a carbon source. Genes on the arabinose operon will basically break down or degrade arabinose. So repression or activation of the arabinose operon depends on the ERA-C dimer. It's a dimer, and it's dependent on whether arabinose is present or absent in the cell. Its function has dual regulatory functions. So when arabinose is absent, the ERA-C dimer is in an elongated form, and that basically represses the expression of the genes that break down arabinose. And that should make sense, because if the whole purpose is to break down the sugar, if the sugar's not there, then you're not going to want to express the genes that break down the sugar. When arabinose is present, then that ERA-C dimer is in a compact conformation, and that will stimulate transcription. Because when the sugar is there, then you want to make the genes to break it down. So the cell is not going to waste its energy on things that it doesn't need to do. So it's very dependent on the presence or absence of arabinose, the reactant in this type of context. So conformational changes of the ERA-C dimer are important. You guys should know, what happens when it's in its elongated form versus its compact form. And so this is just another way of saying it and showing it. And so ARA-BAD is just another way to say the arabinose operon, because B, A, and D are the 3 genes that are on here, but they're not shown. You just show the B. So it's important that you know the ERA-C dual regulator is part of the arabinose operon, and then how it works in the presence and absence of arabinose, its conformational changes, and how that contributes to either repression or activation of the operon. Does that? Yeah.
Could you go over what happens in the A, P, and E sites during translation?
Yeah. So these 3 sites are binding sites for binding sites on the ribosome, and they're binding sites for tRNA. So I'm not going to really question you guys much on this. I think that it's just important to understand that the tRNA coming in to bind the ribosome, it has to bind in certain areas on the ribosome. And so you have an A site, which is an acceptor site. Basically, it's accepting, the ribosome is accepting the incoming charged tRNA. It's an amino acyl TNA because it's charged. The P site is the part of the molecule, the ribosome, basically where it can grow the chain, it adds the rest of the amino acids, and then the exit site is just where it's stripped of the polypeptide and the tRNA exits the ribosome. But I wouldn't worry too much about the details there for this exam. How was the study guide? Was that helpful? When you're studying, I would say like at the end of, like at the very, maybe today or tomorrow would be a good time for you guys to then maybe print out a new study guide and then go through each one and try to recall from, memory and understanding some of the concepts. And then if you find yourself, having difficulty, I would circle those and then maybe go back and then try to understand it. So it's a good tool to kind of test yourselves in your understanding I know some people are like making their own flashcards and things like that. You know, it's not a waste of time, but I think that it could confuse you in knowing what to focus on. The study guide tells you exactly what to focus on for this exam. So if you know everything on the study guide, you should be more than fine. So I would use that as your tool to kind of test yourself rather than maybe doing your own flashcards. Not to say that wouldn't help, like do what works for you, but that's, you know, my advice. Yeah.
Can we go over protein modification?
So after translation, we usually say that at translation, you go from mRNA to protein. It's actually, you know, this protein is made, but it's not a functional protein. It needs to be folded into its 3D conformation. It needs to actually be modified. And so so it's, you know, translation is not the last step in producing that functional molecule. So it has to be modified. And remember, a lot of things have to do with structure. Structure determines function. So those modifications really help it achieve that 3D structure and help regulate its activity. So there's different structures, you know, different levels. You won't need to know this in depth, but some of the things that can happen in terms of this modification or processing is it can cleave that first amino acid, the N-formyl methionine. The others are these 6. Adenylation, phosphorylation, acetylation, methylation. So there's just adding a certain group based on the name to that protein. So that will activate it or just modify it, process it. Protein modification, you know, is basically this type of modification. You're adding something or you're removing that first amino acid, but there's also, you know, other steps of folding and degradation that can happen. Yeah.
So when you remove the first amino acid, what does, like, maybe it's called a neutrophil, what is that?
This was just, you don't need to know this for the exam. This was just an aside. I just wanted to show you guys basically one of the ways that neutrophils are immune cells in our body that help fight bacteria. You know, when you have a bacteria in your body, neutrophils are some of the first responders that try to attack the bacteria. So how do they know the bacteria is there? How do they recognize it? Well, they can sense that part of the protein on the bacteria, which is the N-formyl methionine. So neutrophils basically are attracted to that, that first amino acid that's made by bacteria. But I wouldn't worry too much about that. It's more of like a fun aside. What else? Yeah.
Can you look at the really life cycle of matrix?
Mm-hmm. Three life cycles. Lytic, kill. Like the virus kills the bacteria. Lysogenic, it's kind of like a sleeper cell. The virus is kind of just hanging out and, you know, being replicated with the bacteria. Slow release, the phage is persistently, you know, using the cell's machinery and it's basically replicating itself, but it doesn't kill the bacteria because it needs its cell machinery to replicate itself, so it's kind of taking advantage of it. The lytic cycle, you basically have the phage injecting its DNA into the bacteria. This is the circular bacterial DNA. This is, in purple, is the phage DNA. And so when in the lytic cycle, you know, basically the virus can gain access to the cell's machinery because it's going to make these progeny phage, which are basically the replicated phage from the initial phage that was there. And those progeny phage, once there's a lot of them in there, then they can lyse the cell. Basically, it bursts open, it kills the bacterial cell, and then now we've made, in this case, say like 5 more phage that can go on to infect other cells. So the reason why it's called the kill cycle is because it kills that first bacteria, but then those get released into the environment to go on and kill more bacteria. So it's all about bacterial death. The lysogenic cycle, basically the phage lysogenizes the host, so it leads to a state called lysogeny. So basically what this means is it maintains its genome as a prophage. So here in the lysogenic cycle, a prophage is when you have the phage DNA integrated into the host genome, the host DNA. So in this new DNA, we have basically the phage and the bacterial DNA combined. And then once that happens, it kind of is a sleeper cell because the phage is kind of like, well, I'm integrated into this one's DNA. I don't really need to do anything. So now we'll just allow the bacteria to replicate itself, and I'll just replicate with it. Great. I don't need to kill it. I'm taking advantage. I'm sleeping in this, bacteria's house and just getting all the benefits. The bacteria needs to eat and replicate, so I'm going to be eating and replicating too without it even knowing. So it's like a sleeper cell. And so the key there is that the viral DNA gets integrated into the host DNA as this prophage. The slow release cycle is a little bit different because instead of it integrating its DNA into the bacterial DNA, the DNA gets in there, the viral DNA gets in there, and then it starts to assemble its protein capsid, because remember, every phage needs to have the DNA or RNA genome, and then it also needs to have a protein capsid. So once it's in there, it starts to use the bacterial cell's machinery to make these protein particles in yellow that will eventually become the capsid head for it. But it's using the bacterial cell's machinery to replicate. And so here you see there was 1, now there's 2, it's replicating, and so the bacteria also wants to replicate, and it can, but it does that more slowly because the resources are being used to make phage. Doesn't kill the cell. Lysing the cell means killing the cell, so don't get that confused with lysogenic. Does that help? And also it's important for you guys to know that these life cycles can switch. So you can be in the lysogenic cycle, but then if there's an environmental stressor or something that will threaten survival of the host, then the phage can be like, all right, well, I'm out of here now, gonna wake up and go through the lytic cycle, kill this bacteria, I don't wanna die with it. So that's kind of how it goes. Yes.
Can you go through the half-life of the RNA?
Yes. Where was that? Which chapter?
Chapter 8.
There we are. So several different types of RNA that we talked about. mRNA is the messenger RNA that's made from transcription. That one has a lower half-life, 3 to 5 minutes, which just means that's the amount of time it takes for that molecule to be degraded in half. So mRNA, it gets degraded quickly. Things like rRNA and tRNA, those are the two you should know that have a much higher half-life, which means it takes longer to degrade them. The reason, so half-life correlates with stability. So the longer the half-life, the more stable it is, right? Because if the half-life is shorter, then it's going to get degraded quicker, so it's unstable. And the reason for this is because basically the tRNA and the RNA, they're more stable, they have longer half-lives because they have these modified bases, and the modified basis just keeps them from getting degraded by things in the environment like RNases, which are enzymes that degrade RNA. So you should know that half-life correlates to stability, and you should know which two are more stable than mRNA. So these two. rRNA. Yeah, and just understand why they're more stable, aka they have higher or longer half-lives. Oh, it's snowing again.
Like fat snow. Yeah, outside, fat. It's like slush.
Wow, that just triggered a whole lot of conversation.
I thought we were done with snow.
Well, all I had to say was snow.
We used to have a kid on my school.
Is it though? Is it a spring break?
You're going somewhere.
Bria, did you have a question?
I was gonna ask if we already talked about like protein degradation.
Yeah, we can go through it.
Okay, yeah, I wanna go through that quickly.
Sure. So after proteins get modified, they get folded, and then, it should be a functional protein. And so sometimes things don't get folded properly. Sometimes it gets folded properly, but it's just not necessarily a good, protein, or it's not functional, or something is wrong with it. Things don't always go well. So basically, there are protein degradation mechanisms, basically to degrade the protein if it's not folded properly, or if it's not functional. Those signals that signal that this protein is not functional is called a degron. You don't need to know the N-terminal rule, but what you should know is a protein being degraded. It's the enzyme that degrades a protein is called a protease. So just like I said, RNases degrade RNA, proteases degrade proteins. So another way is in eukaryotes and archaea, it comes in the form of a proteasome, which is just more complex. But you should know endoproteases, that would be the function of it. And then the other thing you should know is that if it's damaged, they don't necessarily always get degraded. They can go into, they can try to get refolded again. There's this triage system. So it can either get folded again, refolded, or it can, and if it's still not repaired, then it can get destroyed and degraded. Okay? I saw another hand.
I was just gonna ask if the endoproteases are the same as the proteases, but I think they are. So.
Yes. Endo just means it's in the cell.
Okay.
Yeah. So it's important to start to remember, some of those roots. So if you see something that ends in ACE, it's an enzyme. It has catalytic activity. An RNase is going to degrade RNA. Protease or endoprotease is going to degrade protein. So keeping those things in mind is helpful when you're reading a question because context is everything. What else? Yes.
Can you go over elongation of transcription?
So elongation of transcription is just when you add the ribonucleotides. So you don't need to know any of this stuff. You should just know, you don't need to know the details. So basically what you should know is that in elongation, you're adding those RNTPs or ribonucleotides. The sigma factor has come off because the sigma factor is required for what?
Initiation.
Thank you. Initiation of transcription. So when we're in elongation, there's no longer a sigma factor. And then there are other proteins at work, DNA topoisomerases, that help to make sure that there's no supercoils ahead so that the RNA polymerase, as it's adding those RNTPs, it doesn't bump into those supercoils. So there's other proteins that kind of unwind those, make sure the DNA is not wound and supercoiled when it's doing its work. That's really all you should know for transcription elongation.
So for translation, termination, so like, I know that like the ribosome complex gets recycled and the protein like detaches, but like is it a stop codon that does this?
Yes.
Okay, yeah.
So in translation, okay, did I miss it? Yeah, basically in translation, the end of translation is when you, oh wait, maybe it was in this one, is when you have stop codons and that signals the end. There we are. Yep, and then completed protein is released, recycles the ribosomal subunits so that they can be used again. You should understand, some of these and how they work. When I write my exams, I literally have the slide up in front of me and I write it, so make sure you pay attention to some of the terms here. Yeah, so for translation, it's just the stop codon that signal.
Okay.
Yeah, and then transcription, you should know the row dependent and row independent, just basic general concepts.
What's the difference between inducer exclusion and catabolite suppression?
Yes, you do. So catabolite repression is when you have, a catabolite is glucose in this case, so it's when glucose represses the lac operon. That's the catabolite repression. Inducer exclusion is basically the process by which glucose can repress the lac operon, and that's by inhibiting the lac-Y, and so it's excluding the lactose from getting in. So that part is called inducer exclusion. So they're part of the same process, but they kind of refer to different things. Catabolite repression is the whole thing. Glucose represses the lac operon because glucose is the preferred energy source, or sugar for energy. And then inducer exclusion is like, how does glucose repress? It's by the exclusion of the lactose. So because if lactose can't enter the cell, the operon can't be induced. So that's where that comes from. Good question. Yes.
So does the reason that it represses it because you already have the glucose, so why make more why use the energy to make more, or why?
So basically, if you have two, both of them, glucose and lactose, present in the media, So bacteria need both, or need some sort of sugar to grow. So like when I make those plates or the broth in lab, I make sure I put glucose in there, you know, some sugar, something for them to grow. It's a nutrient source. So if glucose is the preferred one because basically the bacteria are gonna break it down. They're gonna break down the glucose to make energy. And so when they break down glucose, that glucose can then go into glycolysis, and glycolysis you get ATP energy. So the cell can grow. So once the glucose is used up, then it can turn on genes for the lac operon, which are the genes that are going to break down lactose. So that's why until glucose is used up, then lactose can be used. And so there's no need to, it's not making anything. The glucose and lactose operons basically have genes that break down the sugar. So that's why they're catabolic. So yeah, glucose is the preferred sugar for cells. And so if it's there, it's gonna wait and not break down lactose, because lactose is gonna be broken into galactose and glucose, and then glucose can go into the glycolysis pathway. So that would be why. What else? Yeah.
You just go through that's it.
Yeah. So, quorum sensing, you should know the definition of quorum sensing and the definition of, what was the other one? Chemotaxis. You should know those definitions. And so in quorum sensing, basically when it's a communication mechanism for bacteria. And this is all dependent on cell density. The cell density, or the amount of bacteria, has to be high in order for quorum sensing to be induced. You should understand this general model of how quorum sensing is induced, genes for quorum sensing are induced, and it requires the accumulation of an autoinducer. So when the cells are all at a high density, then they can all produce this autoinducer, and then, you know, it triggers quorum sensing genes to be activated. So bacterial quorum sensing is used both between different species and within the same species. You should know that. You should know why quorum sensing is used for bacteria in just different forms. You know, it can be used to control virulence genes. It can be used for communication. And then, yeah, you should understand that basically this mechanism of what happens first is the autoinducer is produced inside the cell, or outside the cell, and then it diffuses into the cell. So it's kind of like it loops around from going inside the cell to outside the cell. And that's how the cells around each other know that there's this autoinducer being produced by a lot of the cells. So they all start to now do the same thing. They reach a high concentration, and then in the case of like the Vibrio fischeri, they all turn their lights on. So that's their quorum sensing mechanism. We have a few more minutes. What else?
Can you just go through transcriptional control?
it.
Do you know what slide?
It should be in the.
Okay. Okay, transcriptional control. And then any of the right slides? Oh, it's chapter 10. Okay, thank you. Oh, gotcha. So this, yeah, so you just have to know that there are different levels of control for gene expression. So you can control it at the DNA level, transcription, which is what we talked about, and then other mechanisms. We also talked about post-translational mechanisms. But the general concepts of transcriptional control include knowing some of these terms, you know, a regulatory protein, promoter, RNA polymerase, you know, the mRNA. Regulatory proteins come in two forms. They can be activators or repressors. And then ligands influence the regulatory and binding to the DNA sequence. So, you know, that DNA sequence is called a regulatory sequence. So the other thing you should know is differences between DNA or control at the DNA level versus the protein level. At the protein level, it's rapid and it's most reversible. Changing, you know, something or controlling something at the DNA level is going to be the most drastic and least reversible because you're literally going down to the DNA. So if you change the DNA, it's going to change everything else in its process. So control at the DNA level is the most drastic and the least reversible. All right, good luck, and I'll see you guys on Friday.
I might put up in the